So this video is on the topic mitosis. Okay. So mitosis, if we see, is divided into karyokinesis and cytokinesis. Karyokinesis is the nuclear division. Okay. It is the division of the nucleus. Whereas cytokinesis is the division of the cytosol or the cytoplasm. Okay. If the cytoplasm needs to be divided, then the two cells need to be separated. So the cell division is taking this actual cell division is taking place here. Okay. With boundaries. Now this mitosis is divided into uh, two parts: interphase and M phase. First, we will learn about interphase. Okay, so the interphase is again divided into G1 phase, S phase, and G2 phase. G1 phase is also known as gap 1 phase. S phase is also known as synthetic phase or synthesis phase, and G2 is known as gap 2 phase or growth 2 phase. Okay. Now, in the interphase, 95% of the time is consumed in the interphase. Okay, and this is an important. It's an interesting thing that during embryonic growth. Okay, when we are in our embryonic growth, usually in the interphase, the G1 and the G2 is skipped. Like the gap phase is skipped. Only the S phase or the synthetic phase is carried out. Okay. Now, so this thing at early embryonic growth, G1 and G2 is skipped. Only S phase is taking place. Okay. Even though this interface is very biochemically very active, but under light microscope, it is like you can see no visible changes there. Okay. There is a reason to it. The reason is okay, uh, the chromosomes. Okay. They decondense. Decondense means they spread out. फैल जाना, छोड़िए जावा, ठीक है? They spread out and diffuse. Okay, to allow for transcription. Okay, if transcription will happen, then only protein synthesis will happen, then only you can say replication can occur. Okay, because for replication we need protein, right? And how can we get protein? We will get protein if the DNA is getting converted into the RNA by transcription, then RNA is getting translated into proteins, right? So this transcription is very much important. For transcription to carry out efficiently, we need to spread out the chromosomes, right? So the chromosomes are decondensed, they spread out. Okay, and they spread out. Okay, they do their transcription diffuse well, and they do their transcription quite efficiently, and they occupy the territories that needs to be occupied in the nucleoplasm. Okay, now we have discussed about this you can say three phases, right? Now the G1 phase. The G1 phase, here in the G1 phase, you can say cell growth is happening. Okay, RNA synthesis is happening. Okay, we need protein for this S phase, because in S phase we need to synthesize DNA, right? That's a hectic process, and for that hectic process we need much, like large amount of energy. Okay, we need proteins, right? So how can we get those proteins? So we have to first synthesize RNA, then we have to synthesize amino acids, then we can get protein. So RNA synthesis is also seen here, amino acid synthesis is also seen here, and protein synthesis is also seen in the G1 phase. Okay, and ATP synthesis is also taking place because it's a hectic process. So there will be energy investment here. Okay. Second, most organelles, not all. Remember this: most organelles, not all, because there are two organelles that are getting replicated in the G2 phase: mitochondria and chloroplast. Apart from this organelles, all organelles, okay, they replicate in this G1 phase. Microtubule organization center or organizing center or centrosomes. You might have read about centrosomes, that in centrosomes there are two centrioles perpendicular to each other, and aster rays are spread out, and aster means star like. Okay, so under microscope it appeared like a star, so it is known as aster. Okay, aster rays. So these two centrioles along with those aster rays, that is known as centrosome. So here the centrosome, okay, it also gets replicated. Centrosome or microtubule organization center or organizing center. Okay, because you know that from these centrioles spindle fiber originates. From this centrioles or centrosomes, okay, microtubules are arising, right? So this is the reason why it is known as microtubule organization or organizing center, because this centrosome organizes microtubules. They organize the spindle fiber. Spindle fiber is made up of tubulin. Okay. Then chromosomes, here in the G1 phase, chromosomes are highly decondensed and relaxed. Okay. And there is one more thing, that even though this centrosome, okay, it replicates in the G1 phase, but its replication ends at the G2 phase. Okay. And this ends at the G2 phase, but they remain closed in a simple centrosomal complex. It looks something like this. Okay, it looks something like this. Two perpendicular bodies are there, that are known as centrioles. These are asteroids. Okay, and they remain closed in centrosomal complex. So you can say this is centrosomal complex. Okay, now comes S phase. S phase, as we know, synthesis phase or synthetic phase. Okay, so DNA synthesis will happen, DNA replication will happen. So DNA replication or synthesis is taking place. Another thing is that the DNA content doubles, but ploidy does not changes. The ploidy remains the same. 2n remains 2n. Okay, you see this one chromatid. Okay, it replicates its DNA and it gets its sister chromatid. Okay, so the DNA content might have been different, but how many chromosomes are there in total? Here also one chromosome was there. Here also one chromosome is there. Not exactly chromosome, chromatid. Okay, so the DNA content is doubled, but the ploidy remains the same. Okay, no ploidy change is taking place. Now, this sister chromatid, it is encircled by a protein, glue protein. Okay, that is known as cohesin. Now, this glue protein, this cohesin, okay, this protein comes under the protein family of proteins known as SMC family of proteins. SMC family of proteins. SMC. SMC stands for structural maintenance of chromosome family. Structural maintenance of chromosome family. But it is SMC, structural maintenance of chromosome family. So it maintains the structure of chromosomes, because if structure of chromosomes is not maintained, then you will see that tremendous or horrible mutations have been taken place, and the individual is suffering from you can say cancer or something like that. Okay, now at this stage, at this stage also, the chromosomes are still indistinguishable. Okay, they are indifferentiable. Okay, you cannot see clearly visible. These are not clearly visible yet. Another thing is that because the DNA content has increased, so we can say DNA has increased. Now, if the DNA has increased, we need more histone proteins to bind, right? We need more histone proteins to bind. So, histone proteins are synthesized. Histone proteins along with other proteins are synthesized. Okay. So, how is it taking place? Let me give you an example. Okay. If you see, if this is the histone protein, okay, there are 8 such histone proteins. The DNA wraps it like this. Okay. Like this, it wraps it. See, right, tightly, so that the DNA can be coiled. Okay, see, now how many histone proteins are there? Total there are 9 histone proteins. Okay, H2A, H2B, H3, H4. These are here in the core DNA. Okay, the DNA sequence, the thread-like structure, it wraps around these 8 balls. Now you will say only 4 balls are it is visible, right? 4 balls are only visible, because you see, I have highlighted one more thing. So behind these 4 balls again, different 4 balls are there. Okay, other 4 balls are there. Like it is present. For example, this is a ball. Behind it, another ball is there. So when you are looking from front, you are looking that there is only one ball. But if you change the proximity or the polarity, okay, if you change your viewing angle, then you will see that there are actually 2 balls, right? So there are eight histone proteins, and on top H1 is there. So total 9 histone proteins are there. This part is known as linker DNA, and this part is known as core DNA. The linker DNA consists of 60 base pairs, and the core DNA, it consists of 146 base pairs. Okay. Now this, what is this? We learned about the structure. What is the structure? The name of the structure is, this is the structure of the structure of nucleosome. Okay. Now what is nucleosome? Nucleosome is the fundamental unit of DNA packaging in eukaryotic cells. It is a fundamental unit. Like this unit, n number of units, such units are there. Okay. So fundamental unit of DNA packaging. This is fundamental unit of DNA packaging in eukaryotes, not in prokaryotes. In eukaryotes. Okay. So nucleosome is present in eukaryotes, not in prokaryotes. Fundamental unit of DNA packaging, right? And nucleosome is this histone octamer. Okay, 1, 2, 3, 4, and another 4, 8 histone octamer plus DNA linker and DNA core. Now, how many wraps are there? This DNA wraps uh, 1 whole 3 by 4th time, okay, or 7 by 4 if you multiply this into this plus this, 4 into 1 is equal to 4 plus 3, 7 by 4, or you can say 1.75 because 1 and 3 by 4 is 0.75 its value is. So we can say a complete round, then 3 by 4th of the round, okay. And then comes G2 phase. Here in the G2 phase, similarly how this G1 in the G1 phase cell growth was happening, in G2 phase also cell growth will happen. Okay. In G1 phase, RNA, amino acids and proteins are being synthesized. Here also RNA, amino acid and proteins will be synthesized. A protein named tubulin will be synthesized here. And as we said that most organelles, not all organelles replicate here in the G1 phase. Similarly, the organelle that was left behind, mitochondria and chloroplast, will duplicate or replicate here. Okay. Now, if the protein is being synthesized, so there will be more transcription. Keep this in mind. Okay. And here then it is ensured that there is no DNA damage. Okay. Then it is pushed into mitotic phase or M phase. That M phase we will discuss in the next video.